So we ended our last lecture looking at the lysosomes. Lysosomes are membrane-bound digestive vesicles arising from the Golgi apparatus. Basically, they contain the enzymes that are going to catalyze the breakdown of macromolecules. They contain those hydrolytic enzymes. They also destroy cells or foreign matter that the cell has engulfed via phagocytosis. Lysosomes themselves have a pH of about 5 to 8, which help play a role in intracellular digestion. There are also structures called microbodies. Microbodies are a variety of enzyme-bearing membrane-enclosed vesicles. A type of microbody is peroxisomes. Peroxisomes contain enzymes involved in the oxidation of fatty acids. Hydrogen peroxide is produced as a byproduct, but we know that the cell counteracts the hydrogen peroxide, which acts as a poison to our cells and can destroy them by producing an enzyme called catalase. And you know, catalase, when breaking down hydrogen peroxide, forms water and oxygen gas. Vacuoles themselves are membrane-bound structures in plants. They have various functions depending on the cell, the type of cell involved. There are different types of vacuoles. The one that you are commonly familiar with is the central vacuole found in plant cells. The central vacuole itself is surrounded by a membrane called the tonoplast, and it's going to store things. There are also contractile vacuoles that help osmoregulate. Protist, also found in some fungi, and you have storage vacuoles. Here is a picture of the central vacuole of the plant. There you can see the membrane surrounding the central vacuole called the tonoplast, and that's going to store nutrients and water for the plant cell itself. Mitochondria, they are found in all types of eukaryotic cells. They are bound by membranes, an outer membrane, and then an inner membrane, and then you have the inner membrane that is folded to form cristae. Between the outer and inner membrane, there's an intermembrane space, and then of course inside the inner membrane, the bulk volume would be the mitochondrial matrix. On the surface of the inner membrane, there are proteins that are going to help carry out oxidative metabolism. There are a lot of enzymes that are going to help play a role in part of aerobic respiration. Mitochondria are pretty neat because they are structures that have their own DNA, mitochondrial DNA. And actually, mitochondrial DNA is actually inherited from your mother. So it's something that you could thank your mother for on Mother's Day, because she has given you her mitochondrial DNA. So if we look at the mitochondria itself, here is its structure. You could see the cristae, you could see the circular mitochondrial DNA. There are ribosomes in there, inner membrane, outer membrane, the intermembrane space, and of course that inner membrane is folded to form cristae, and ultimately that is going to increase the surface area for certain reactions of aerobic respiration. The chloroplast is another organelle that is present in the cells of plants and some other eukaryotes, like blue-green algae. It's going to contain pigments, especially the main photosynthetic pigment chlorophyll, which is green. It too is surrounded by an inner and an outer membrane. And then inside you have sacs called thylakoids, and the thylakoids are going to be the location of the light-dependent reactions for photosynthesis, because that is where the pigments are going to be stored. A stack of thylakoids is referred to as grana. Like mitochondria, chloroplasts also have their own DNA. So basically, when we think of the structure of the chloroplast with its DNA, and also the mitochondria, its structure with its own DNA, we also think of them as once free-living organisms. So here's a better look at the chloroplast. You could see the thylakoids, the stack of thylakoids called granum. They too have their own ribosomes, their own DNA. So that brings us to this idea of endosymbiosis. And basically what endosymbiosis or the endosymbiotic theory basically proposes is that some of today's eukaryotic organelles evolved by a symbiosis arising between two cells that were each free living. One cell, a prokaryote, was engulfed and became part of another cell, which was the precursor of today's modern eukaryotic cell. An example would be like the mitochondria and the chloroplast. So there you can see a nice example of the unknown archaean, protobacterium, and a cyanobacterium all coming part of the modern day eukaryote. The cytoskeleton is, as it suggests, the skeleton of the cell. It is a network of protein fibers found in all eukaryotic cells. It is used for support, supports the shape of the cell, and it also keeps organelles in fixed locations. What's really neat is that unlike our skeleton, the cytoskeleton can reform itself. So we could say it is a dynamic system, constantly reforming and disassembling its structure, which is basically composed of three types of fibers. You have those fibers that are called microfilaments, which are the actin filaments. Microfilaments are two protein chains loosely twined together. Movements like contraction, crawling, or pinching are going to help in those movements there. You also have microtubules. These are the largest of the cytoskeletal elements. They contain dimers of alpha and beta tubulin subunits. Microtubules are going to facilitate in the movement of cell and materials within the cell. So when we think of it serving as that monorail system, it is going to be those microtubules that are going to serve in that manner. There are also the intermediate filaments. Intermediate filaments are between the size of actin filaments and the microtubules. They are very stable structures, and they are usually not broken down. If you look at the overall structure of the cytoskeleton, here you can see the actin filaments, microtubules, and the intermediate filaments. In the animal cell, there are things called centrosomes. Centrosomes are regions surrounding the centrioles in almost all animal cells. Microtubule organizing center. They can nucleate the assembly of microtubules. So centrosomes are regions surrounding the centrioles of most animal cells. It's a microtubule organizing center, can nucleate the assembly of microtubules, and basically we know that animal cells and most protists have centrioles that are going to play a role in cell division. Plants and fungi usually lack centrioles that are going to play a role in cell division. So there you can see the microtubule triplet. So you get microtubules in forms of 3, and ultimately altogether you have 3 to 9. So it's 1, 2, 3, 4, 5, 6, 7, 8, and then 9. So you get that 3 to 9 complex there. Cell movement is essentially all cell motion is tied to the movement of actin filaments, microtubules, or both. Some cells crawl using actin microfilaments. You also have the flagella and cilia, and flagella and cilia are going to have a 9+2 arrangement of microtubules, which is separate from that of the prokaryotic flagella. The cilia are shorter and more numerous than a flagella itself. Cilia look like a bunch of little hair projections that are going to extend off the cell, where a flagellum is going to be 1. Some cells might have 2 flagella. If you think of the tail of a sperm cell, that would be representative of flagellum. So there you get that 9 to 2 complex there for flagella and cilia. There are also special structures in the eukaryotic cell. For the plant cell, we have extracellular features like the plasmodesmata, and you have a cell wall. In the animal cell, animal cells don't have a cell wall. They are going to have an extracellular matrix. So the eukaryotic cell walls found in plants, fungi, and many protists is different from the cell wall that is found in prokaryotic cells. The cell wall here is going to be made up of cellulose. So plant cell walls and the cell walls in protozoa are going to be composed of cellulose. In fungi, the complex carbohydrate there is going to be chitin. If you go back to thinking of the cell wall of the prokaryote, the cell wall of the prokaryote is going to contain or not contain peptidoglycan. Plants have both primary and secondary cell walls. The secondary cell wall may or may not be present, but it does have a primary cell wall. The primary cell wall is going to be laid down. If we're talking about a plant, it will be cellulose. So you have the primary cell wall of a plant cell here. Here's the primary cell wall of an adjacent plant cell. And then in between, you have a space called the middle lamella. In the animal cell, you have the extracellular matrix. Animal cells lack those cell walls. They secrete an elaborate mixture of glycoproteins in a space to surround the cell. Collagen fibers may be abundant as well. Ultimately, this is going to form a protective layer over the cell surface. Like the integrins are going to link the extracellular matrix to the cell cytoskeletons. And ultimately, it's going to influence the cell's behavior. So if you look at the extracellular matrix, here you have collagen fibers, elastin fibers, proteoglycan, integrin proteins, fibronectin, and ultimately you're going to have that integrin protein that's going to connect the ECM to the inner side of the cell's cytoplasm. So there's an overview of cell structures and their function. We also have things that do cell-to-cell interactions. Cell-to-cell interactions is going to play a role in cell recognition, cell communication, talk about how cells are going to adhere to each other. So cells make contact and can read each other and react. Glycolipids are cell-to-cell interactions. Most are tissue-specific cell surface markers. You also have MHC proteins that is going to serve as recognition of self and non-self cells by the immune system. That will play a role in whether a cell should defend itself or not. You also have cell connectors. 3 categories of cell connectors based on function. You have those that are tight junctions, those that are anchoring junctions, and those that are communicating junctions. In a tight junction, you're going to connect the plasma membrane of adjacent cells in a sheet that has no leakage. So a tight junction will be representative of the cells that will make up the tissues of the stomach, for example, because you don't want, they act like zippers because you don't want any leakage of the stomach contents into other parts of the body, as it could lead to severe infection and possibly death. Anchoring junctions are mechanically attaches the cytoskeleton of neighboring cells. Examples of anchoring junctions are desmosomes. And then you also have those communicating junctions, which are going to allow for chemical or electrical signals to pass directly from one cell to an adjacent cell. In the animal cell, we call those communicating junctions gap junctions. In the plant cell, we call those communicating junctions plasmodesmata. The plasmodesmata are going to be found in the primary cell walls of adjacent cells. So they're those little channels that are going to allow things to pass through. So here you can see an example of tight junctions, anchoring junctions, and communicating junctions. Plasmodesmata are specialized opening cell walls. Cytoplasm of adjoining cells are connected. So here you can see that channel between one cell and the other. There's the primary cell wall. Here's the primary cell wall of another cell, and there's that middle lamella. So the plasmodesmata extends across, allowing for cytoplasmic joining. The function is similar to a gap junction in the animal cell itself. So that is it for cell structure function. It concludes our chapter 4. Thank you for watching, and if you have any questions, please feel free to ask in class. Have a great day.